Modern Blood Banking and Transfusion Practices, 6th edition, Chapter 3, Fundamentals of Immunology. All red blood cells have many antigens present. If cells are transfused from an allogeneic blood donor, they will have antigens that are not the same as the patient's. These antigens present on transfused cells can elicit an immune response and antibody production. Our job is to prevent, detect, and identify the antibodies. Antibodies against red blood cell antigens are found in the plasma and are very dangerous as they can cause transfusion reaction or hemolytic disease of the fetus and newborn. In blood banking, we are primarily looking for and detecting alloantibodies. We can also detect autoantibodies that are made against the patient's own red blood cells. We don't look for autoantibodies against other parts of the body in blood bank. Auto means self and allo means against others or foreign antigens. An antigen is a molecule found on the surface of a foreign cell or a damaged internal cell that the body may react to and make antibodies. B cells, when stimulated, become plasma cells or memory cells. The plasma cells are the antibody-producing cells. Immunoglobulins or antibodies make up a high percentage of the total protein in disseminated body fluids, about 20% in a normal individual. Antibodies are classified by their molecular structure of their heavy chain. IgG makes up about 80%, IgA about 13%, IgM about 6%. The inset shows formation of the Fab and Fc fragments after enzymatic cleavage of the IgG molecule by the enzyme papain. Most clinically significant antibodies that react at body temperature, or 37 degrees Celsius, are IgG isotypes and are capable of destroying transfused antigen-positive red blood cells, causing anemia and transfusion reactions of various severities. Most IgG antibodies, like the one shown, can react at immediate spin and can fix complement via the classical pathway. The complement system is a complex group of over 20 circulating and cell membrane proteins that have a multitude of functions within the immune response. The primary roles include direct lysis of cells, bacteria and envelope viruses, as well as assisting with opsonization to facilitate phagocytosis. Opsonization just means like coating the cell to kind of flag it for the immune system to pay attention to. The complements sections were ordered in the order they were discovered, not the order of activation. Without calcium and magnesium, the pathways cannot be initiated. When using plasma samples, the complement in the plasma cannot be activated. Notice that the classical and alternate pathways have points where they are both the same. They both lead to the membrane attack complex, which is called C5b6789. A fragments have anaphylaxitoxin activity. They promote inflammation and they mediate immune responses. The lectin complement pathway is activated when mannose-binding lectin is attached to microbes. The membrane attack complex, which is C5b6789, is the goal of all three pathways, as it can lead to complement mediation, cell destruction of bacteria, enveloped viruses, and cells. This is very dangerous if it occurs to transfuse red blood cells. Complement can bind to red blood cells, and in the transfusion service, we work to prevent this from happening. However, sometimes patients can have antibodies that are too low of a titer to detect. The patients might have no history of antibodies being detected in the past, and you can end up with a reaction. This can occur due to activation by IgG or IgM antibodies. IgG-Rh antibodies are particularly dangerous, while ABO antibodies are deadly. Antigen characteristics influencing immune responses are size, complexity, conformation, charge, accessibility, solubility, digestibility, chemical composition. The different blood group antigens differ in their immunogenicity or ability to cause an immune response. Polyclonal means they are heterogeneous population of antibodies. So that means more than one, uh, or antibodies to more than one different epitope. A single antigen with multiple epitopes will elicit multiple antibodies to it. So you could be sick with a bacteria and make multiple antibodies to different parts of the bacteria. Monoclonal antibodies are reagent antibodies that have been created by isolating individual B cells from a polyclonal population. Naturally occurring antibodies can be produced without transfusion, injection, or pregnancy. For example, anti-A and anti-B antibodies are naturally occurring. Most are IgM, react at room temperature or lower, activate complement, and may be hemolytic at 37 degrees Celsius. These can include the ABH, HI, Lewis, MN, and P blood groups. Immune antibodies are produced due to exposure to the antigen. This could be by transfusion or pregnancy. Miscarriages and abortions can still lead to antibody formation if the mother is exposed to the antigens from the child. These are IgG and react at 37 degrees Celsius. Most require anti-human globulin for detection. These are a problem for Rh, Kell, Duffy, KIT, and S blood group antigens. Unexpected antibodies are detected through the indirect antiglobulin test or an antibody screen. They are unexpected because they should not normally be there. We have naturally occurring ABO group antibodies, and we must have methods in place to detect the antibodies to prevent transfusion reactions. The host factors that influence immune response include nutritional status, hormones, genetics, age, race, exercise level, disease, and injury. It is important to know that reactivity of naturally occurring ABO antibodies declines with old age and is not present at birth but develops shortly after. One of the most important steps in obtaining the correct and valid results of an analytical or diagnostic test is to have the correct sample. Misidentification of samples can lead to the death of the patient. Most of the time, plasma samples in pink top tubes are used, but if we need complement to be active, then a serum tube must be used. The method used for testing a blood bank is hemagglutination. This means that the red blood cells agglutinate. A precipitation test is when a solid is created from a solution. Agglutination inhibition is when we block a reaction. Hemolysis is lysing of red blood cells. Because hemolysis is a positive reaction in blood bank, we cannot accept samples that are hemolyzed. The tube test uses hemagglutination and is the gold standard for blood bank testing. It has two phases, sensitization when the antibodies attach to the red blood cells and lattice formation when the sensitized red blood cells link together to form a clump. Centrifugation is an effective way to enhance agglutination reaction because it brings reactants closer together. The zeta potential is the natural repulsive effect that prevents red blood cells from getting close to each other. A prozone is when you have a negative or no clump because there are too many antibodies present. And a postzone is when there are too many antigens. Both lead to false negative tests. There are several factors that affect the reactivity of antigens and antibodies. The pH should be 6.5 to 7.5, but a few antibody reactions are enhanced by acidification of serum. These are anti-M and anti-PR. Temperature is especially important in differentiating antibodies. IgM antibodies react optimally at ambient temperatures, and this is called immediate spin, while IgG antibodies react at 37 degrees Celsius. The type of antibody will react differently and require different techniques. IgM works well in saline, while IgG works better with colloids or low ionic strength saline solutions. We use enhancement media when we do antibody screens to detect IgG antibodies. There are multiple types of antibody enhancement medias. List reduces zeta potential. PEG increases the dielectric constant and reduces zeta potential. Enzymes enhance some antibodies and eliminate other reactions. Anti-human globulin reagents allow the lattice formation to form on sensitized red blood cells. There are differences in manufacturing techniques for reagents. Polyclonal reagents are directed against multiple epitopes found on the original antigen used to stimulate antibody production. These could be called human reagents. They used to collect plasma from donors who had been exposed and created antibodies. Monoclonal reagents are directed against specific epitopes. They can cause overspecificities and may miss some antigen if a patient has a slightly abnormal version that is missing that epitope. Complement may not be fixed, or you may detect antibodies that are not clinically significant. This means they react in the lab, but not in the body.